All right. Yes. It's always worth a shot. All right. If I'm not mistaken, and then here's where we left off the last time. And was describing the 6 different types of neuroglial cells and then their individual functions, leaving off with what I think is probably one of the more important ones, which is the oligodendrocytes and Schwann cells because of their effects on myelination. So again, purpose of myelination was to speed the transmission of the action potential. Get it from point A to point B a whole lot faster so that you can survive. You don't get the 3rd-degree burn because you put your hand on the stove and it has to travel every inch of your arm. Right? It will jump, get to the brain quickly, you move your hand back. Right? Because that is not an involuntary action. It requires the brain to interpret its ought to move the hand away. Okay. All right. Well, that was the function then of the neuroglial cells. Who's the actual one transmitting the electrical impulse? Well, that would be the neurons. So the neurons themselves are the ones who actually are the ones transmitting the action potentials. You remember that whole joints, structural, functional? Same thing applies here, right? There's an anatomy that they have that determines their physiology or function. Right? So there are 3 types of neurons based on, again, structure and function. Functionally, we're going to kind of do this backwards from what we did with joints, because with joints we said, here's what their structures are, fibrous, cartilaginous, or synovial, and then here's how they function. Don't move, move a little, move a lot. Right? So the functions that we have are either sensory, motor, or kind of both. Okay? Inter ones are the ones that connect. And the best way I think of this is if I'm driving downtown, I'm going to get in my car, I'm going to go on I-10 going east. Right? I don't just then put the car in reverse and come back on same I-10 on the same side of the street, right? I got to cross over and get on the other side of I-10 to come back. Well, that interneuron is the connecting to get from one side of the street to the other, right? So sensory is going one direction, motor is going the other, but we do have to have a connection between those. That is the interneuron. Okay? So if I touch a hot stove, what's going to go up my arm? Sensory. Well, the neuroglials are going to transmit it up there, hopefully fast, but that's the sensory going up. Then I'm going to transfer over and then send a motor back down to move the hand away. All right, so those are the 3 functions. Sensory, motor, or kind of a both, a transition between the 2. Okay. All right, when it then comes to structurally, I have 3 classifications. Multipolar, bipolar, and unipolar. And I'm actually going to kind of describe these in opposite order from that. I'm going to go unipolar, bipolar, and multipolar. Okay. But what does uni tell me? 1. 1 what? Well, 1 polar. Okay, but in the context of a neuron, there were arms and legs coming off of these. Axons and dendrites. Uni tells me what? 1. There is only one of those things coming off. Okay? So, what always does a neuron have? An axon. So, if it's unipolar, it means it has 1 axon and 0 dendrites. Because I have a total of how many things coming off of it? 1. Bi tells me 2. So, I'm going to have 2 appendages. Well, I know one of those has to be an axon, which then I have to have 1 dendrite. What does multi tell you? Any or multiple. So what do I have? 1 axon, and then 2 plus dendrites. So again, the similarities they all have is they all have to have at least 1 axon. The number of dendrites can be variable. Okay. So in picture A, unipolar. B, bipolar. C, multi. D, there's only one thing coming off, and then it splits. I don't care the split. I need to know how many things come off that main body. Right? So you can think of them as arms and legs. So it's attached to the body. Well, in this case, only one thing, and then it branches. I don't care about the branches. So that indeed is unipolar. Okay? So again, we have a combination of dendrites and axons. Now, the dendrites are like arms. So, and this is physiologically, again, speaking, kind of think their function. They're going to bring things in. The fancy word for that is afferent. Afferent, afferent, again, a number of different ways to describe it. I kind of make them sound grammatically different. So I go afferent and efferent rather than afferent, efferent, because then they kind of sound alike. So, afferent means in or towards. So, in this case, there is no dendrite, but let's say this one's a dendrite, so the action potential is going towards the body. Right? And do you bring things in with your legs? No, you can, but usually you're doing what? Kicking out and pushing away. So, efferent means away. So, axons are sending the information away from that neuron cell body. Now, in reality, I would have to see the impulse to know under the microscope which one is truly the axon, which one's truly the dendrite. We just said, pick the wider one, that's the axon. But in reality, you need to know which way the impulse is traveling. Okay? When we bundle all of these together, they're going to create a nerve. So going up and down your leg is a sciatic nerve. Up and down your arm is an ulnar nerve. Okay? Those are going to have a combination of axons and dendrites just wrapped together. Okay? Some of those are going to be carrying sensory up, some of those are going to be carrying motor down. So, which ones are carrying what? Okay? So those are the structurals. The functional was sensory, motor, or both. Multipolars carry motor information. Okay, just put the M's together. Multipolars, motor. The unipolars carry sensory. I know it's going to seem a little tricky. What's left behind is the one that's kind of connecting, so it's kind of the both. So the inters, you'd say, are the bipolars, and that seems reasonable with their because these are also sensory. The interneurons could be any of them. And so what's the difference then between these two? We'll eventually get more detailed in this. But the first are general sensory. The second are special sensory. So do you remember the difference between connective tissue proper and specialized? Connective tissue proper versus specialized? One was like adipose, the other was bone. Adipose was connective tissue proper, bone was specialized. Blood was specialized. Right? What was different? Can I find it in multiple systems or just one? So what makes a sensory general? It's all over the place. As opposed to special, where it's only one location. So I only smell with one place, the nose. Right? So that neuron is going to be a bipolar because it's a special sense. Pain, on the other hand, can you feel only pain on your foot? No, I can feel pain on my abdomen, my heart, at multiple locations. So that's going to be a general sense type of a neuron that carries pain. Okay. Now, again, if this is the leg, not good artwork, it's kind of messed up jokes down there, but the idea is it's not meant to be again good artwork. Going down the leg is going to be your sciatic nerve. So when you step on a rusty nail, okay, what's going to take that pain action potential up your leg? A unipolar neuron, because that's a general sensation, that's going to carry it up the leg. Your brain's going to interpret, I think I just stepped on something sharp. What should I probably do? Pull my leg up and away. So we're going to then send motor back down that same nerve, down a multipolar. Wrap them together, we just created that sciatic nerve. So it's a combination of sensory going up, motor coming back down, but on different neurons. Okay. All right, so eventually again, know the details as far as what's carrying what, and we're going to eventually piece all of this together. And we're going to start by stepping on a nail, following the action potential through its course. Okay. All right. So the job is to carry an action potential. What is the action potential? It's an electrical impulse. It's a current. Think of it like a lightning bolt. Okay? That is the action potential. You know this, you just don't know that you know this. It is not carried in the cytoplasm. It is transmitted along the cell membrane. Do you remember that whole neuromuscular junction? Started with the brain, sent it down to the neuromuscular junction, crossed the synapse. Then what happened? It started an action potential on the sarcolemma, got taken down into the T2. What I didn't do is the same thing is actually happening on the brain, sending it down the neuron. Okay? If this is, again, the neuron cell body, and here's the axon, okay, and we're going to get down here to that neuromuscular junction, we're not traveling down the cytoplasm, we're traveling down the cell membrane. When I finally get down here, calcium comes in, acetylcholine gets released, transfers over, and then we're now traveling on the sarcolemma, right, on the cell membrane. So it's going to travel along the cell membrane, not down the middle of the cytoplasm. And again, I got right, then right, then right, then right, then right, whatever. That current, again, is an electrical charge. Think electrons. When we're thinking charged particles, what's the fancy name of those? Ions. So what's going to happen? We're going to move ions, which are charged, across this cell membrane. Again, you know this, you just don't know that you know this. At that neuromuscular junction, okay, calcium came in, acetylcholine got released. What happened on the other side? We opened doors for Na plus. Okay, plus tells me it's a ion. What came in was Ca2 plus. That is a ion. Okay, so again, it's the ions, the positively and negatively charged ones, that are going to then create this current. Think of it like electricity. Yes. Okay. Now, to do that, once again, was this calcium just in and out at all times? No, there had to have been a door for it, or a channel, another fancy name, right? What was down here? Channels for sodium. Those channels are what are going to allow those electrons or ions to move.
So whenever they deliver a shock in CPO, is it thing where they like you're just doing the same thing? You're trying to create the flow of it.
Correct. You're trying to basically send like a signal from the brain would have come from, but is now just an extraneous outside source of a current. Yes, to open up the channels.
Does it do diffusion, like the calcium and sodium ions, like do they also do?
Yes, but what kind of diffusion?
Simple diffusion.
No, facilitated.
Facilitated, right? Because they are charged, even though they're small, they are charged, which requires they go through an integral protein. Now again, that was topics we've already talked about. Nice, good question though, because it keeps it fresh. Comprehensive final exam, it's like a month away. Can you believe it? 4 weeks. One of those weeks is Thanksgiving, so we're like 3 weeks away from your final.
No.
Don't be frightened. Okay, think of the positives. Right? All right, so when it comes to the channels, okay, when it comes to the channels, we have two types: leaky and gated. So the leaky channels, or non-gated, means those channels are open at all times. Okay, so almost kind of think, and you live in Houston, you can appreciate this. There are subdivisions or communities that are non-gated, which are leaky, anyone and everyone can drive in. And then there were others where there is a gate and a security guard there, which you have to have open, right? So then that would be the gated channel. Right? So the leaky ones open all times, the ions are constantly moving. The gated ones, the door is closed. Something's gonna have to cause that door to open. We have one of two types. It can either be voltage gated or ligand gated. Okay. So what does voltage tell you? If I have a voltage, I have a charge. Right? So to open this gate, the ions themselves, an electrical charge is gonna cause that to happen. And I'm gonna give you a very prime example here in mere moments.
Like a clicker, like you give a clicker for that.
This is me driving up to my garage, hitting the clicker from a distance, sending an electrical current to the garage door opener and it magically opens. Okay. Ligand, on the other hand, is me having to get out of my car, walk over to the door, grab it, and open it. Right? Actual molecule has to attach to that door to cause it to open. That is called ligand gate. Okay. So in this fancy picture here, right, this gate is closed. So I see negatives and positives. So that's a charge which causes that gate to now open. So they were closed, they're now open. So that one is voltage gate. As opposed to this one, that gate is closed. This molecule attaches and opens it. That is now ligand. So, brain starts in action potential, travels down. What happens? Ca2 plus came in. That was a gated channel, because it wasn't open at all times. If it was, calcium would constantly be coming in. So it's closed. What caused that door to open? The electrical current. So that is a voltage gated channel. Caused what? Acetylcholine to cross and attach to receptors, which then opened sodium channels. So what caused that one to open? An attachment. So that is now ligand gated. Now there's little videos, and I usually know who watches the videos, because this is their answer when I say, please describe the events of the neuromuscular junction. Their answer goes something like, an action potential starts in the brain, travels down an axon, reaching the axon terminal, causing voltage-gated calcium ion channels to open. I never say that. Now we know the voltage-gated. Causing acetylcholine to get released in the synapse. Acetylcholine crosses the synapse, attaches to acetylcholine receptors located on the motor end plate, causing ligand-gated channels to open. So again, the difference is that one was due to a charge, this one required a molecule attaching to it. The events of the neuromuscular, yes. Do you have to include this being voltage gated and this one being ligand gated? No. Okay. But still that calcium comes in, causes release of acetylcholine, calcium receptors, open sodium channel. That, yes. So, who are my ions of interest when it comes to an action potential? Now, calcium again was mainly for muscle contraction, right? So, this electrical current is due to what? Sodium and potassium. And so, Na plus K plus. They are both ions because they're charged. So the two ions of interest for action potentials are sodium and potassium. All right. Finish that. Go ahead.
Other than do they need to be charged to be ions of interest? But
Yes.
They would be an ion.
They would be ions, but they weren't charged. Correct. Now again, some will say, well, where's calcium? Again, calcium primarily think has to do only with muscle contraction. Right? Action potentials need sodium and potassium. All right. So let's look at the details of an actual action potential. And to do that, we're going to break it down into 3 steps. Resting membrane potential, depolarization, repolarization. And then what we're also going to do then is relate it to this graph. Okay. So I'm going to repeat my little graph, and we're going to keep coming back to it for important kind of details. This is basically time on the bottom, and this is charge. So period 1 here is considered resting membrane potential. Going up the hill is 2, which is depolarization. And then going back down the hill is 3, which is repolarization. Now this picture I realize has a step 4, which is this undershoot. That's still kind of as the pill is going down, so we're going to attach that to repolarization. We're going to list this as 3 separate steps. All right, now for this to work, okay, we're going to have my neuron cell body, dendrite, and then an axon coming away. What I'm going to do is magnify that section of the axon. Okay. So we're going to have a cell membrane on one side, cell membrane on the other, cytoplasm with organelles floating around on the inside. So if you want, you can put a mitochondria in here, some rough ER, some smooth ER, some lysosomes. Just realize that's cytoplasm. Again, that's not of interest to us because our action potential is traveling on the cell membranes. What's going to be along this cell membrane? Channels. Some of these channels are going to have open, okay? Others are going to have closed. And now, I'm going to assume this is not myelinated. Okay? Because if this were myelinated, an oligodendrocyte or swan is going to cover all of that, which is why we're going to jump over it. Right? So let's assume this is not myelinated, so all of this is visible. Okay? So this would be a leaky channel. This one is gated. And again, it's going to happen on the bottom as well. I'm just making it visible higher up so that hopefully everyone can see. Okay, but it's basically the same picture down at the bottom. Okay? All right. For this to work, think of it like your car battery. All right? Now, some of you are like, I don't know much about cars, but at least hopefully you know something about car batteries, that there is a positive terminal on one side and a negative terminal on the other. Okay? And this is how our action potentials are going to work. Okay? We're going to have a charge on one side that is opposite to the charge on the other. That's our border. Just like our battery, we're going to have positive on one side, negative on the other. All right. What does this first thing tell me? Is there an action potential currently being sent? No, because it is at rest. But it has the potential. So, is your car currently running in the parking lot?
No.
Probably not. But it has the potential, assuming you have a functional battery that has a positive and a negative terminal. Okay, so the first thing I need to have for this to even work is the possibility that I have a positive on one side, negative on the other, that I have a functioning battery. But it is currently at rest. Okay? Then what's going to happen? We're going to depolarize, which is now going to take us out of rest and go up the hill. Okay? Let's look at the word. What does polar tell you? Right? Remember our whole covalent bonds? We had non-polar and polar covalent. Kind of opposites. So I have a positive and negative. So what's going to happen? Depolarize, we're going to remove or take it away. Right? If I dehydrate, I take it away. It's kind of a misnaming of it, I guess. We're not going to remove it. What we're going to do is switch them. So at rest, we're going to start with this. What we're going to do when we depolarize is switch them. So it's a little bit of a misnaming, because depolarize kind of means to remove it. Right? We're not really removing it, we're replacing them, okay, or switching them. But then look at the last one, repolarize.
This one.
I'm going to then return them back to what they were. So we're going to have rest, depolarize, and then repolarize, which returns us back to rest.
So it'd be like if you're, when you start your car, right, it has like typically like 3 clicks like until it starts on. So for the ignition, you can say that that's like 1, 2, 3, and like once it hits the back, the repolarizer starts.
Sure. I don't know if I had a line. It's an okay scenario, but
You get what I'm saying?
kinda. There's a delay, obviously. It's not an instantaneous, but
Yeah, it's like tick, 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 and on.
yes,
Not
yes,
exactly that noise, but you know.
yes,
that.
yes. All right. So we vaguely get an idea of what's happening. Okay. So let's describe rest first. So at rest, there is not an actual potential. However, possibility exists. Why? Because I have polarity. There's positive on one side, negative on the other. So at rest, who is located where, and what charges are located where? So at rest, and I'm going to get rid of this top picture because that's kind of now that's in the way. Now that we know what we're magnifying, okay? At rest, who is located outside? Sodium. Who's located inside? Potassium. Na plus A plus. Now these are both pluses, and I know what you're thinking. I thought we just said we had a plus on one side and a minus on the other. Okay, and that is true. Again, we have a high concentration of sodium, Na plus outside, a high concentration of K plus inside. The net charge outside is positive. Why? Because we have Na plus. That makes sense. The inside at rest is negative. And it's more to do with the fact that what else is in here?
Other stuff.
Other stuff. In addition to the potassium, who, yes, happens to be positively charged, but it's due to this other stuff that the inside is negative. Okay. So, on my picture, my starting point here at rest is negative. I don't care that you know it's negative 70. Just the fact that it's negative is important. So, net outside positive, net inside negative. And again, the negative is because of the other stuff that's in there. Now, I'm not saying there's no sodium inside. I'm not saying there's no potassium outside. I'm just saying the higher concentrations are outside for sodium, inside for potassium. Now, why do I mention that? Remember a little thing called homeostasis? Do we like things to be high on one side, low on the other? No, we want them to be equal and balanced. So is there a way we can do that? Sure, we've got channels that these can now move through. So, at rest, there is movement of ions. However, we're not going to change the charge, because what happens if my battery loses the positive on one side, negative on the other? It ain't going to start. Right? I need to maintain positive and negative. So do I want these to move? No, I don't want them to move. I want to keep positive outside, negative inside, but body says we want to try and get these to homeostasis. So are they going to move? Yes, because I have higher on one side, lower on the other. And oh, look at that. There's channels that are always open that allow them to move. So, Sodium is gonna move in through a leaky channel, because it's higher outside, so it's gonna leak inside. Potassium is higher inside than it is outside, so what's gonna happen to the potassium? He's gonna leak out through a leaky channel.
Is that the facilitated diffusion?
So that would be a facilitated diffusion. Now, I'm not gonna ask that on this test. You're like, why? I know that. Comprehensive final, perhaps. So again, sodium moves in through leaky, potassium moves out through leaky. Again, do we like that? No, because then we're gonna lose my positive and negative. Everything's gonna get to equal. So what do I have to also do? Have a little merry-go-round over here. Okay. That pumps the sodiums back out that leaked in, and pumps the potassiums back in that leaked out. What do we call that? The sodium potassium pump. Anyone recall what kind of method of transport? It's active transport. Okay. So the sodium potassium pump was active transport, because I'm going to go from where it's lower to where it's higher. Because the sodium's higher outside, it leaks in, and we're going to pump it back out from low to high. So the comparison I give here is, I'm in a boat. My boat happens to have holes in it. So can I stop those holes from letting the water in? No, I can try. Okay. But what do I have? I have my little coffee cup. Okay, so as the water leaks in, what do? Scoop it out, and I keep throwing it overboard. So the sodium comes through, and I just then take my coffee cup, swap it back out. So it's a constant movement. But what does it allow me to keep doing and maintaining? Sodium outside, potassium inside, with my positives and negatives being maintained. Okay. So even at rest, do I need energy? Yeah, because this ATP requires ATP energy pump, right? To then keep moving that back out to position. So while we're here, keep it fresh. Okay. What charge is outside? And we're talking rest only. Trying to keep all this stuff. What charge is outside? Positive. Oh, that's the answer to my apologies. So positive. Charge inside. Negative. Ion outside. Sodium. Ion inside. Potassium. Is there any movement of ions, and if so, through what channel? Yes. Leaky channels.
Pump.
And the pump. You can see it now. Do we even have to mention? In a few words of your own, please explain the resting membrane potential. Your answer goes something like, well, in resting membrane potential, there is currently not an action potential. However, I have a positive charge outside due to sodium. I have a negative charge inside, not due to the potassium, but that's where potassium is located. Due to the other protein sulfates, but a high concentration of potassium inside. And the ions move through leaky channels and the pump. All right, so that's kind of that initial key parts. Okay. That explain at least level one.
That's just
Rest. Say again?
That's just level one.
That's just level one. Now the good news is, it's not going to get super complicated, because we already said all we're going to have to do is switch these, and then switch them back. Okay. And how do you think we might do that? What have we yet to discuss or describe? The gate. All we're going to do is open gates and let them move. Okay. Now, we don't stay at rest at all times. Right? We have to then depolarize and start an action potential at a certain point in time. Right? And so, do I currently need a action potential for pain being sent up my arm? No, because there's no pain stimulus. But if I touch a hot stove, I better do what? Send an action potential to my brain that says, hey, your hand is painful, you better move your hand away from the stove. So I can't stay at rest. We gotta reach this dotted line. With that, then indeed is threshold. Okay, so remember that for muscles? Does a muscle just randomly contract? No, it has to reach threshold for those fibers to contract. Same thing, what's gonna cause me to leave rest is if I have a stimulus that meets threshold. So, same term, same overall concept. Alright, so if we reach threshold, we're going to leave rest and now depolarize. Okay. So in depolarize, we're not going to lose it, in fact we're just going to reverse it. To do that, sodium gated channels now open. Where's sodium high? Outside. So what's going to happen when gates grid open? It's now going to go inside. And what charge does it have? It's Na plus. So what's going to happen in the inside? All these pluses are going to come in, so it's going to change the inside to plus. And if all the outside no longer has pluses, it's now going to become minus. Okay. So the only thing that happens in depolarization is the sodium gates open, allowing sodium to now flood in. Potassium gates stay closed. The leaky channels are still leaking. The pump is still pumping. Those are not changed. The only thing that changes is I open doors for sodium. Now, basically, I still got my little coffee cup, okay? I still have the little holes that are seeping in, but now I got these big holes that are allowing the water to just flood in. Can my little coffee cup keep up? No. So that's why this overwhelms this and changes it to now positively charged. So what happens? We don't stay at negative. We're now going to go up the hill. And do we go to 0? No. What's my final point here at the peak? Positive. Now, do I care that it's positive 20 or positive 30? No, I just care that it's a now positive charge inside. Okay. So again, here it is kind of represented. The leaky channels are still leaking. The potassium gates stay closed. The sodium gates now open, allowing all the sodium charges to come in. So the inside is now positive, the outside is now negative. And I don't know that I would ask this, but I guess I could. At this point, top of the hill, who's inside?
Sodium.
Sodium.
And potassium are now both inside. Who's outside? Basically nobody. They both are, but in very low levels, right? Because it's not as though they 100% are in one location or the other. But more of them are now both inside. Now, at the top of the hill, what's also going to happen? The sodium gate now closes and traps the sodium inside. Okay, so the door opens, allows the sodium to come in, and then closes. Traps the sodium now inside. Now, if it were up to me, I would have designed the system in such a way to then, to repolarize, do what? Open the sodium doors, allow the sodium to go back out. Unfortunately, that's not how it works. Okay. We're kind of with me so far, though. Smile and nod. All right, so the net result is what? The inside is now positive and the outside is now negative. So we're going to go back down the hill to get us back to rest. As I already mentioned, it would have been nice just to reopen the sodium gates and allow the sodium to go back out. Unfortunately, that doesn't happen. So instead, how can I return this back to normal? Well, I have another item that happens to be positively charged that I can send back outside to get the outside back to being positive. So what are we going to do in repolarization? Swap it out. Let potassium out. So in repolarization, we're going to return back polarity. Right? So in this case, the potassium gates now open. Potassium, who is now positively charged, floods out, carries with it a positive charge. If all the positives inside are now flowing out, the inside returns to being negative. Leaky channels are still leaking, pump is still pumping. Those are not changing. The only thing different is now potassium gated channels open. The sodium gated ones again are closed. The leaky channels in the pump still functioning.
So at the rest period, both the pump and the leaky channel are working to
Correct.
keep the homeostasis.
To maintain positive outside, negative inside. Correct. To maintain the homeostasis, no action potential sent, to maintain polarity. Correct.
So during the whole entire duration, the pump and the channel, the leaky channels remain.
Always functional.
Always functional.
So again, in this case, leaky still leaking. Now we open the potassium gates, that floods out, changing the outside back to positive, the inside back to negative, and the sodium gated one stayed closed. The only thing that would make this picture better is if they put all the sodiums on the inside, right? Because they already came inside the last picture.
So what's stopping them from being backwards? Like say that it was potassium first and then sodium second, you're still repolarizing and depolarizing regardless of.
Theoretically, yes. I didn't design the system. I don't have an answer to that.
Okay.
Kind of like, why don't I just reopen the sodium channels and let the sodium back out? I mean, it's one of those where there's multiple to the answers, like why did.
Okay.
I don't have.
You can think of
So second, I'm sorry.
you can think of the sodium channel is higher than fluid work. It's time for the potassium channel to work. They have a sheet. It's different curve.
Yes, but again, to his question, why did they open the potassium first and allow the potassium out to make, again, I don't.
So like if we were to put that on like a question, I mean technically it's not wrong, because you're still repolarizing and depolarizing regardless of the, like, who comes first.
No, well, yes and no, because again, the goal is the outside was already positive, the inside was already negative. If I open potassium and allow potassium out, that's gonna just take more positives with it.
So it needs to be completed.
So it has to be able to reverse. The positive charge that's outside, to bring that in, I have to open the sodium one first. So yeah, in hindsight, that can't go the opposite direction.
I'm saying that with potassium is outside first. and
No,
sodium is
that,
outside.
exactly. If potassium were outside first, then yes, I would open potassium channels first, you would think theoretically, because that would create the scenario I need.
That's what I'm saying,
Reverse
so
the
you can't,
charges.
like if you can't be flopped out at
You
will.
can't, yeah,
Okay.
yeah. All right, so the end result of repolarization is that we return our charges back to normal. Right? Outside is now positive, inside is again negative. So right here at the bottom of the hill, we're once again back to a negative charge, and what's going to happen, the potassium gates now close, trapping the potassium outside. Are we back to rest?
No.
We are currently right here. Where's rest? Rest is that line. I am currently down here. Right? So although my charges are correct, positive outside, negative inside, I still have some problems. And I'm not currently at rest. Because at this point, who's outside? Potassium outside. Sodium's inside. That's not rest. Because at rest, sodium is outside, potassium's inside. Rest is this negative 70. I'm now actually below that. So I've got actually two problems. Seems like everything was kosher, but we actually have two problems that have been created. Right? The charges, agreed, have been returned to normal. Right? The problem is the ions are not located in the right spot. Because at rest, sodium was outside, but right now, sodium is inside. In addition to that, the potassium-gated channels opened for a longer time period than the sodium ones did. So I'm gonna exaggerate this, but let's just say that door opens for 5 seconds. How many students can come in? I don't know, 3. If that door opens for 10 seconds to let students out, more can actually go out than came in because it was open for longer. All right? So in that case, again, sodium and potassium weren't open for the same amount of time. The potassium one was open for longer, allowing more potassium to go out. And once again, potassium has a positive charge, so more pluses left, which meant the inside became more negative. So we're not negative 70, we're now like negative 90. Negative 100. Again, I don't care the actual number, it's just more negative than what I should be at rest. So this is what, in this picture, they're calling the undershoot. It's still technically part of repolarization, right? Because the gates don't close until the bottom of the hill. So it's not a particular extra step, okay? But that's what we're going to label as either the undershoot or what's also called the after potential. Now everyone got that previous line. So again, ions are located in the wrong spot and charges aren't really correct either. We're more negative than we should be. All right, so this again is termed the after potential or the undershoot. Once those potassium gates close, all I need is time. Because where's all the potassium right now? Outside. So in a leaky channel, which way is it going to now leak? It's going to leak back in because it's going from higher to lower. And what's the pump been doing all along? It's already been doing that, right? Because the sodium was leaking in, the potassium was leaking out, the pump was already pumping it back in. So all I need to do is say, don't let any more channels open, and me and my little coffee cup will eventually get there. Right? I'll just start scooping it all out since no more water is flooding in. I just need time to get me back to rest. So once the potassium gates close, the leaky channels on the pump will return ions to their proper location. They're going to leak and get pumped the direction they need to go. And it's not an equal exchange. Right? I pump 3 sodiums for every 2 potassiums. Again, eventually that'll take care of this more negative charge. You don't need to know that detail. Just know that all I need is time to let the pump and the leaky channels do their thing. And that will return me back to both charge and the ion locations. And so we are technically back to rest. But again, from here to here is going to take, now we're not talking 20 minutes, this is still milliseconds, but it's still, I need that little brief pause to give the channels and the pump time to do their thing. All right. So in a few words in your again, basically just be resting membrane potential. Okay, to describe what ions are located where, what charge is located where, what kind of channels and movement of ions is occurring. And then I, in a multiple choice, could ask you, in depolarization, what channels are open? The leaky and the pump. In depolarization, the sodium gate. Now technically these aren't channels, but I want you to include the pump. Okay, because that's causing ions. Alright. So again, this kind of shows you, I think they use plus 30 instead of my plus 20. I don't care. Right? It's knowing that we're starting off at a negative charge. What's the dotted line? That would be threshold. If we meet that, we will then depolarize, finishing up at a positive charge, going back down to the negative, resting gets after potential undershoot. So once the neuron depolarizes, from that moment until I return to resting membrane potential, that period of time is called a refractory period. Okay. From the moment I leave rest until I get back to rest, that is termed a refractory period. The refractory period is then broken down into an absolute and a relative refractory period. Okay? So, the lighter blue here and the lighter blue there is rest. The really light blue, okay, is your absolute refractory period. The darker blue here is then your relative refractory period. So, both the lighter and the darker is a refractory, broken down into an absolute and a relative period.
So absolute would be the finite stuff and change, and then relative would be with decompression.
Yes. So at peak versus depression. Another action potential cannot ever start during an absolute period. So, during this period of time, can I start another signal? No. From here until there. Once I reach this point, do I have to wait till I get here to start another? No. If it is a supramaximal stimulus. Now this goes back to our muscle. What were my 5 kinds of stimuli? Threshold, submaximal, maximal, supramaximal. So if it is a supramaximal stimulus, I can start another action potential during this period. If it's not supramaximal, I will wait until I get to that point. Now, some of you can mentally picture this. The description I'm going to give you, others again, maybe not so much. This is again after the fact. I always think, because I always usually forget to bring the Twizzlers on the day that we describe like the month 5 years. This year I happen to remember. But then I always think, oh, I should remember to do this. So, at the gym, I've seen this. They've got this really long rope attached to the wall, and you sit there and you kind of do this with the rope and create little waves. One kind of mentally picture that. Maybe you haven't been to the gym and you're like, yeah, I don't know what you're talking about. But maybe you've seen it on TV. Okay, so they go up and down, creating the waves. Right? So the idea is if I lift up and pull down, that's going to create a wave that's going to go towards the wall. Right? Can I start another wave on top of the one that I've already started? Is the question. Right? So can I go, and start a second one? The answer is no. You have to wait until that wave has gotten to a certain point before you can start another wave to move along the current. Okay. If, however, it is supramaximal, body's not going to wait. Right? If it's supramaximal, we're going to say, you know what? We're going to start another action potential before we get to that point. Because this is such an intense signal you need to know about, brain. Okay? If it's not, we will wait till we get there and then send another action potential. So, the general period from rest to rest is called a refractory period. And so, a refractory period, as its definition is I cannot start another action potential during that. Unless it's a supramaximal, in which case during the relative portion of that, we can. But only if it's supramaximal.
So would there ever be a diagram like that on there? So at that, it's just doing another group.
What can you say about this one? It stayed at rest. Never met threshold. Right? Never sent an action potential. It just stayed at rest. What can I say about this one? Well, we went through the entire hill, so we went through the entire refractory. Right? And then we sent another signal, and then sent another signal. So is that just a threshold level stimulus? No, we probably would have only sent one. Right? So this would be either submaximal or maximal. Right? We're sending several signals. What do you say about that one? We didn't wait. We're like signal, which means that is a super maximal stimulus. So again, maybe it's seeing the picture that way kind of helps.
Yeah.
Okay. Now, what do you notice about those latter two pictures though? The heights of those are always the same. Little thing called all or none. So, hopefully you can appreciate not all levels of pain are the same. There's different levels of intensities. That's that whole submaximal, maximal, supermaximal. Action potentials, however, just like muscle, are either all or none. If I send an action potential, it goes from that negative 70 to the plus 20. It makes no difference if I'm being pinched or if I'm being stabbed. Those are two different levels of pain, right? Yet, the brain receives a action potential that was negative 70 to plus 20. So it's not as though, let's see if I can focus on this little section. For what I'm describing here, it's not as though a pinch, and here's our action potential. Oh, it's a pinch. Okay. Oh, this is a stab. No. That's not what happens. It's all or none. So if I send a current, it's going to have the same height each and every time. So how do I know a pinch versus a stab? The stab, which is a much higher level of pain. There you go. We're going to send more frequent action potentials. So the brain receives the signal, and how many it gets, the brain will then interpret as how intense that pain is. Right? So the stronger the stimulus, the stronger that the pain is, the hotter the object is. Right? The brain determines that based on how frequent the action potentials are. So those two are not the same level of pain. What's that one? That one's being stabbed. What's this one? Being stabbed, burned, and shot. All at the same time, right? Super maximal.
So, my brother actually, he's doing a clinical for paramedic. He got to bring home one of the EKGs and a patient who was was burned alive, who was dying.
Yeah.
It was 192 beats per minute, and that was exactly what it looked like.
Yeah. Yeah.
Yeah.
Because the heart's like, I gotta keep going for this. Because again, it's such a shock at that stage. Yes. Yeah, it's like, I can't wait. This is intense pain. I need to keep sending signals. Okay. But again, every signal that's sent, same strength. Brain just interprets the information, and from that says, okay, this is really intense. Now, there's a big part of that. Right? Is all pain the same in every person? Two people can both get stabbed, and one of them can say, what's in 1 out of 10? What's the pain? He's like, it's a 4. The other can be like, oh my God, that's a 10. Right? That's the brain's interpretation of the information it received. So many ultra pains. Yeah. If I had answers for that. Alright, so to understand this, I'll give you a couple of concepts. What's the difference then between being pinched and being stabbed? The frequency, how many action potentials the brain actually receives. And then obviously the interpretation part of that is the second part. What's the difference then between putting your hand in 300 degree water and 500 degree water?
It's going to be the response.
Is there? Could you be blindfolded and say, 300, 500?
Not sure. Know the difference between hot and hotter.
No.
At that point, you're maxed out. Right? 212 is boiling, first off. So 300, it's already boiling water. So technically 500 degrees is hotter than 300. But if you put your hand in boiling water and even hotter boiling water, you're not going to be able to tell the difference. Because, again, there is a maximum number of signals I can send. Right? During an absolute, I can't start another one. I got to wait till I get to that relative. Now again, I never did point that out. But why can I do it during the relative? Let me go back 2 slides.
The relative, because it's like going back to your
What's magical about that point right there?
There's a threshold on
It's back to the negative 70. Up to this point, I was positive, and negative. Now I finally got to negative 70. My normal resting, at least charge. Even though the ions are in the wrong location, at least we're at the right charge. So we can send another action potential at that point if it's super maximal. But again, 500-degree water, even though it's hotter than 300, is already maximal. So can I start another one here? No. So even though 500 is hotter, you're maxed out. And that's what I'm trying to get at with this. What's the difference between 300 and 500? 500 is technically hotter, but you just can't send any more action potentials. They're already both super maximal. Yeah, that there's going to be a point where, you know, I kind of gave that scenario. Again, stabbed, on fire, and shot. It's probably no different than just being on fire. Right? It's the same level of pain that your body's detecting. You're not having a higher level of pain, even though that's clearly more intense. Okay? All right, so a little final reminder. It is the number of action potentials that reach the brain that the brain interprets that strength of stimulus. Okay? Also keep in mind, smaller and larger number of receptors may be affecting the interpretation part of this. Now let me give you an example of this. Now that's why I like the pool water over here. Okay? If you're like, I think it's a beautiful day, I want to go swimming today. What's probably the first thing you're going to do before you jump in? Check it, and by doing what? Put your foot in there. Right? Like, oh, that doesn't feel so bad. You jump in. Does it feel the same? Why does it feel way different than what it just felt like with your foot 2 seconds ago? Did the water magically get a whole lot colder and the 2 seconds took you to dive in?
No.
No. It's just now your entire body is receiving that temperature signal instead of just your foot. So you're sending signals from everywhere to the brain, so the brain's going to interpret that way differently. Right? So if I have a little drop of oil come out of the pan and land on my arm, and I go, ouch, that's going to feel way different than the entire pan falls on me. Right? Even though it's the same hotness of grease, it's just more surface area, so my brain's going to interpret that way different. Right? So, again, there's a lot, and this gets really complicated in the details, so I'm just trying to give you a basic understanding. All right, I won't get that today. Stop there. I only got like 2 slides left, so we're almost done. Meaning, because I always forget to mention this too. On Thursday, we'll finish up test 3 content. All right. We will likely move on into test 4 content. Okay, so download, print, save, whatever you want to do with those next group of notes to have you those with you if you wish to have those again for Thursday's class. And so we've got, like I said, 2-3 slides left on this content as well. Okay. All right. Today's a big day, so I get more break. Look at that, 1 minute. Get